Hey everyone, Miss Calabrese here. Welcome back. In this video, we're going to be talking about the blood system. All right, so first of all, blood is connective tissue, but it's a fluid connective tissue, and the main job of blood is to serve as transport. So it moves things around the body. So in addition to transporting things like your hormones, which we talked about in the previous lectures, it's also an important mechanism for transporting nutrients, transporting wastes, and transporting gases around your body. Um, it's also going to be an important part of your defense system, so protecting you from pathogens, parasites, things like that. Your blood is a key component of that defense system. And then, in addition to all those things, your blood also helps to regulate your temperature. So parts of your body that are warm, we can regulate blood away from those and towards parts of your body that are colder to help maintain a stable temperature. All right, so what's blood made of? So it's got both a fluid component and a formed element component. That would be like the solid component, the cells. So plasma is the fluid component. The formed elements are the actual blood cells. So plasma accounts for about 55% of the total blood volume, whereas the cells account for about 45%. And as far as how much blood you have in your body, this is dependent on body size, sex, and things like that, but somewhere between 4 and 6 liters of blood at any given time. All right, here's a breakdown of what blood is actually made of here. So again, about 55% plasma, 45% formed elements. Within that plasma, we see things like proteins and other solutes, but that plasma is mostly water, right? 91% water, and then we've got these other proteins and dissolved substances in there. And then for the formed elements, we can divide those into our three major categories of cells. And those are the erythrocytes, which are red blood cells, leukocytes, which are white blood cells, and platelets. Okay, so here's just a kind of graphic so that we can see what different blood compositions look like. So the test tube there shown in A, that's representing a regular blood distribution where we've got, our 45% formed elements and our 55% plasma. Condition B is what anemia would look like, where we don't have enough of those red blood cells, so we don't have enough iron carrying, that's the component of hemoglobin that's carrying oxygen around in our blood. Test tube C there represents a condition known as polycythemia, which is having too many red blood cells, which is a cancerous condition. All right, so let's talk about the plasma specifically before we get into the formed elements here. So again, plasma is that liquid part of blood. It's about 90% water and 10% solutes, solutes being the things that are dissolved in the water. Those solutes a lot of those solutes are going to be proteins. So the plasma proteins are going to be really important for things like maintaining blood viscosity. So that means like the thickness of the blood, so it's not too thin, not too thick, as well as the blood pressure and blood volume. All right, so here's what these plasma proteins are. So albumins mostly serve as transport proteins for lipid-based molecules that have a hard time traveling around in a liquid medium. So albumins would be the type of transport proteins that carry around your steroid hormones. Remember, we said that they need protein carriers in the blood. That job is done by albumins. Globulins are broken down into a few different types, so alpha, beta, and gamma. Alpha and beta globulins also serve as transport proteins for lipids and also your fat-soluble vitamins that need to travel around in the bloodstream. So vitamins A, D, E, and K are fat-soluble, which means they're going to use these globulin proteins to help transport them around in the bloodstream. And then the last type of globulin there is a gamma globulin, and gamma globulins are also known as antibodies or immunoglobulins, so that's important for your immune system. All right. And one more type of protein that we find in the blood is fibrinogen. Fibrinogen is going to be an important protein for the clotting process, but in this form that we have named here, fibrinogen, it is currently a dissolved protein, so not clotting. All right, and then the formed elements we mentioned before are three types of cells. So erythrocytes are red blood cells, thrombocytes are called platelets, and then leukocytes are white blood cells. So getting into what red blood cells look like, what they do. So these are some of the smaller cells that you have in your body. They're in this kind of donut shape, so biconcave disc. They don't have a lot of the same internal components that a lot of your other cells have. So upon reaching maturity, they get rid of their nuclei. They don't have mitochondria, ribosomes, or other organelles, so they kind of get rid of all of that to the point where the primary component of the inside of these cells is just a molecule called hemoglobin. And that hemoglobin is going to be really important for carrying oxygen. So that's pretty much the entire contents of your red blood cells once they reach maturity, because they've gotten rid of a lot of those other important cellular components. Okay, so yeah, the main function of them is to carry oxygen around in the body. They also can carry carbon dioxide, although they don't carry it in the same way that they carry oxygen. And the oxygen carrying is far more important than the carbon dioxide function here. So again, hemoglobin is the name of that molecule that they have that they use to transport oxygen. So each tiny little red blood cell you have has somewhere between 200 and 300 million molecules of hemoglobin per cell. So it's a lot of protein molecules in that single cell. Hemoglobin is a quaternary protein. Excuse me. So it's a protein that has multiple three-dimensional subunits to it. Each of the 4 subunits that hemoglobin has contains a heme group. So that heme group is an iron-containing group. And it's the iron-containing group there that is able to chemically bond with oxygen so that it can carry oxygen around in your bloodstream. So when hemoglobin binds with oxygen, then we refer to it as oxyhemoglobin. Like I said, it can also carry carbon dioxide. If it's carrying carbon dioxide, we refer to it as carbaminohemoglobin. But again, that's less common than the oxygen carrying component. As far as amounts of hemoglobin, males tend to have a little bit more hemoglobin than females in their blood cells for I don't know what reason. All right, here's what that hemoglobin looks like. So again, I mentioned we've got 4 three-dimensional subunits here of this quaternary protein. So in each one of these subunits, the different chains of the protein are going to have their own heme component, and that heme component is where we find the iron. And each one of those can carry 1 molecule of oxygen. So and 1 per subunit. So that means every full molecule of hemoglobin can carry 4 oxygen molecules. All right, so how do we make red blood cells? It's through a process called erythropoiesis. All blood cells are going to be made in your red bone marrow. So if you can remember us talking about that when we talked about the skeletal system last term, bone marrow is where we're making all of our blood cells, so that's where hematopoiesis happens. But erythropoiesis is the specific term we use for the formation of red blood cells. So basically, we take some stem cells. So we have what are known as hematopoietic stem cells in the bone marrow, and those hematopoietic stem cells are going to divide and differentiate into other types of cells, which eventually will differentiate into red blood cells. And this is a process that's constantly happening. So you're making 200 billion of these every single day. Every single day you're making 200 billion red blood cells. So that's a lot. And that's because you're also killing off 200 billion every day. So many are being born, many are dying. And so it leads us to this kind of balanced number of red blood cells. All right, so the destruction of red blood cells, when I say many are dying, This happens roughly every 3 to 4 months. A red blood cell dies. Well, no, many red blood cells are dying all the time, but each one lives about 3 to 4 months. Um, they when they when they've reached that age, then just become eaten up. So they get phagocytosed by macrophages, which are just kind of roaming around the bloodstream, looking for the old ones, looking for the ones that have started to become abnormal, are the ones that are starting to break down. So those ones are identified as being um, uh, old and are marked for destruction. So they'll be ingested by a macrophage and then broken down into their component parts. So we'll break down the hemoglobin. So we'll have a protein component of that, which will break down into individual amino acids, which get recycled. And then we have this bilirubin component. So this is gets recycled, sent to the liver, and used to make bile. Here's what this hematopoiesis process looks like. So way at the top of the picture here, we can see that multipotent hematopoietic stem cell. So, and that just means that that particular stem cell is capable of becoming all of these different type of blood cells, right? So those are the type of stem cells that you have in your bone marrow. So when that stem cell divides, he's going to divide along one of two lines, either the myeloid stem cell line or the lymphoid stem cell line. The myeloid stem cell line will continue to diverge and differentiate, and that line will end up forming a whole bunch of different types of cells. So everything from your platelets to your red blood cells, and to many of your white blood cells, so basophils, neutrophils, eosinophils, and monocytes. Okay, so those are all cells that come from the myeloid line. The lymphoid stem cell line doesn't make quite as many cells, but it's capable of making your natural killer cells, as well as your two different types of lymphocytes, your T and B lymphocytes. All right, so let's familiarize ourselves with a feedback loop that controls the number of red blood cells in the body. All right, so we've talked a lot about negative feedback loops already this week, so let's add another feedback loop into our repertoire here. What's going on in this figure is that we've got a person who's been exposed to less oxygen availability at a higher altitude. So this is what happens if you go way up in the mountains. If you're very high above sea level, the proportion of oxygen in the atmosphere up there is lower. And because there's less oxygen availability in the atmosphere, you have to be more efficient about getting that oxygen to your cells. So what will happen is you'll actually produce more red blood cells. Right, so as we have fewer or we have less oxygen circulating in the bloodstream, your kidneys are going to be the first one to notice that. So your kidneys are really good at noticing the amount of oxygen availability in the bloodstream. When they notice that we've got a low oxygen, they're going to produce a hormone. That hormone is called erythropoietin, or EPO. Erythropoietin then travels through the bloodstream and finds its way to the bone marrow. When it finds its way to the bone marrow, it's going to trigger the production of more red blood cells. So we'll get some production along that myeloid stem cell line to result in more erythrocytes. And then as more erythrocytes are released into the bloodstream, we're more efficiently able to carry oxygen with more cells. And that's going to turn off the feedback loop so that we don't need to create any more EPO because now we have an adequate number of red blood cells. All right, so moving on to the white blood cells, or the leukocytes. So these leukocytes, we have several different types of them. We're going to break them down into two major categories, and those are going to be the granulocytes and the agranulocytes. So granulocytes are white blood cells that have large granules in their cytoplasm. So in this picture here on this side, you're looking at a neutrophil. Neutrophil is a granulocyte, and you can tell it's a granulocyte because it just looks grainy, right? We can see that grainy appearance in the cytoplasm. Neutrophils are the most numerous white blood cell you have. So at any given time, about 65% of your white blood cells are going to be neutrophils. These are highly mobile cells. They're extremely phagocytic. and they are capable of a process called diapedesis. Diapedesis means that they can leave the cardiovascular system and go into the tissues. So they can kind of leak out of a capillary and go into tissue. Right. Another type of granulocyte here, pictured on the top of the slide, is an eosinophil. So an eosinophil, we have far fewer of these, so only about 2 to 5% of your white blood cells are eosinophils. We find them a lot of times in close to mucous membranes, so in the lining of your respiratory tract and your digestive tract, we find a lot of eosinophils. Um, they are good at processing allergic reactions and parasitic worm infections. So if you ever find yourself unfortunately infected with parasitic worms, your eosinophils are going to be part of your first line of defense against that kind of infection, and they're also going to be important for fighting or for initiating an inflammatory response, especially for allergic reactions. And the last one listed here, the last granulocyte we'll talk about, is basophils. Basophils are your least numerous white blood cells, so they can be kind of hard to find on a blood smear just because there aren't all that many of them. These guys are also highly mobile and capable of diapedesis. The granules that they contain in their cytoplasm have both histamine and heparin. So these guys are important inflammatory mediators as well. So if they release histamine, that's going to trigger inflammation, itchiness. If they release heparin, that's going to be a blood thinner. So these guys are important for mediating the inflammatory response as well. Right, and then moving on to the A granulocytes. So two types of A granulocytes here, the lymphocytes, of which we have two types, your B lymphocytes and your T lymphocytes. So these guys are important for your specific immune response. And then monocytes, which are your super large white blood cells. So these are the biggest white blood cells. They're incredibly phagocytic. Their job is just to swim around and eat bad guys. All right, so as far as numbers of white blood cells, 1 cubic milliliter of blood has anywhere between 5,000 and 9,000 leukocytes in it, and it's important to be sort of familiar with that number because there is a clinical significance to that, right? If you have fewer than that or more than that, that's something that your physician would need to know about your health. And then as far as formation of these cells, it's the same hematopoietic process that we mentioned before, right? So we've got the myeloid stem cell line and the lymphoid stem cell line that both come from that multipotent hematopoietic stem cell that we find in the bone marrow. All right, just to show you real quick what this diapedesis process looks like, since a few of your white blood cells are capable of this. This is an important part of your immune response, and again, we mentioned that blood is important for keeping you safe, for defending you against pathogens, and this is part of how that works. So in the picture on the top there, we see damage, right? We see a break in the skin, and that break in the skin has allowed some pathogens entry beyond your first line of defense. So these pathogens are now in the skin tissue. And so what happens is the cells that are nearby where that damage has occurred are going to basically send out some distress signals. So they're going to send out some chemical signals that say, hey, we've got some damage here. We're being invaded. We need some help. That information, those chemical signals are going to drift into nearby capillary, and it's going to trigger the diapedesis of some of these cells. Right, so we're going to be able to, those cells are going to be able to kind of leak out of the capillary. So the eosinophils, the neutrophils, and the monocytes are then going to be able to leak out of that capillary and into the tissues and do their job. Right, so the eosinophil is there. He's able to release these cytotoxic granules that he has. The neutrophil is going to be able to do phagocytosis and eat the bad guys, and the same thing is true for that macrophage. I just want to point out there that the name of that particular cell changes. So a monocyte, it's called a monocyte when it's traveling in the bloodstream. After it undergoes diapedesis and we find it in the tissues, now we call it a macrophage. right? So two different names for the same cell. All right. And then a quick mention about our formed elements, or I'm sorry, our last formed element, which is the platelets. Platelets, also called thrombocytes. These are not full cells, so they are fractions of cells that form from kind of a shattering process of a larger cell in the development process. So these are very small. Somewhere between 150,000 to 400,000 per milliliter is a normal amount of platelets in the blood. And again, these are for clotting purposes, right? So their main job has to do with clotting, which is stopping the flow of blood. All right, so yeah, so their main role is in hemostasis and coagulation. So hemostasis means stopping of blood flow from an injured vessel. So I want to point out that I'm saying hemostasis and not homeostasis. Right, so homeostasis is the maintenance of stable internal conditions in the body. Hemostasis means literally stopping blood. So um, hemo means blood. Stasis means stop. So we're just stopping the flow of blood. And your platelets are an incredibly important part of that process to stop the flow of blood if there's been damage to a vessel. All right, so there are a few different ways that platelets help us in that hemostasis process. So they can form what's called a platelet plug, which is what happens when a bunch of them aggregate together in order to stop the flow of blood. And if the injury is really extensive, then they can also trigger the coagulation pathway. So we'll talk about that here in a second. All right, so how do we form platelets? The specific word for platelet formation is thrombopoiesis. So we're making between 2,000 and 3,000 platelets from every large mature megakaryocyte. So a megakaryocyte is the name of the cell that platelets come from. So this is one of those steps in the differentiation pathway between the stem cell and the final step, which is the platelet itself. Platelets don't live long at all, so these guys they live to be about 7 days old before they begin to break down and then are phagocytosed. All right, so here's our steps of hemostasis. All right, so if there's been damage to a vessel, all right, so say I give myself a paper cut or something, the first thing that's going to happen is known as vascular spasm. So the actual blood vessels, there's contains smooth muscle in the walls of those blood vessels. That smooth muscle is going to contract in response to injury. So you'll get this actual vascular spasm where the vessel itself tries to contract to prevent blood loss. The next step is the formation of the platelet plug. So this happens when we do damage to a blood vessel. You're basically exposing the underlying connective tissue that's holding that vessel together. When that underlying connective tissue, specifically collagen fibers from that connective tissue, are exposed into the bloodstream, they trigger the platelets that were just happily floating through the blood. They trigger them now to, instead of being regular platelets, to become what we call sticky platelets. So they become a little bit spikier, stickier, and they start to stick together. And that's how we develop this platelet plug. So it's just a response to exposed underlying collagen fibers that happen when damage has been done to the lining of a vessel. Right. And then the third step in that hemostasis process is coagulation, which is the actual clotting of blood. All right, so coagulation is incredibly complicated. We're going to try to keep it pretty simple here and just talk about it in the context of two basic pathways here. So the extrinsic pathway and the intrinsic pathway. The extrinsic pathway is when the clotting process is initiated by chemicals from outside the damaged blood vessel. The intrinsic pathway is when the factors are present inside that blood. All right, so extrinsic comes from outside the vessel, intrinsic starts on the inside of the vessel. Okay, so here's what part of this pathway looks like. So I want you to notice one of the terms here, fibrinogen. It's right in the middle of our pathway. We mentioned that term fibrinogen early on when we talked about the presence of proteins in the plasma. So fibrinogen is one of those plasma proteins that's just sort of floating around, dissolved in the plasma, not causing anybody any trouble until we start one of these clotting pathways. There are a few important factors here. So prothrombin is one of our clotting factors that's going to help initiate this response. So when prothrombin is created either from the extrinsic or intrinsic pathway, we are going to get this catalyzing enzyme thrombin created. Now thrombin is going to do something important to fibrinogen. So fibrinogen, we mentioned, is a dissolved protein in the blood, but once that fibrinogen interacts with thrombin, it becomes a different type of protein called fibrin. So thrombin essentially converts fibrinogen to fibrin. Right now, fibrin is not a dissolved protein. So now this protein comes out of solution and starts to look kind of like a web or a mesh. So it no longer is dissolved and flowing. Now it's kind of standing out and getting stuck in the way. And so that's how we begin to form a clot, as now we've taken this dissolved protein, taken it out of solution, and it starts to clump together. So here's what that looks like. So we can see here we've got damage to the blood vessel here. As we've got damage to the blood vessel, we're exposing underlying collagen fibers. That's going to trigger the platelets in the area to become sticky, and we'll begin to form this platelet plug here. But that's only part of the process, so that platelet plug helps. But then we also need to initiate our clotting process. So prothrombin catalyzes thrombin, which is going to convert fibrinogen to fibrin. And then that fibrin, as you can kind of see here, looks like a web, looks like a mesh. And so what it's going to do is help kind of lock those thrombocytes in place. It's also going to grab any other red blood cells, white blood cells, any other stuff that's just floating through the blood can get caught in that web and become part of the clot, right? And that's going to stop the loss of blood through that damaged hole that was created. All right, so conditions that oppose clotting would be, for example, a perfectly smooth surface on the inside of your vessel. So, and that's what we want normally, right? So normally we don't want clotting to happen. Clotting to happen when you don't want it can be really, really bad because we want blood to flow. We want blood to flow all over the body and get where it needs to go. So a perfectly smooth surface is one way of preventing unwanted clots. And then there are also antithrombins, which are circulating in your blood that help prevent the thrombin from converting fibrinogen to fibrin. Conditions that would hasten clotting is any kind of rough spot in the endothelial lining of a blood vessel. So we want that inside lining of the vessel to be super smooth. If it's not, if there are rough spots, then that can trigger the beginning of a clotting process. And those rough spots can come around. You can cause damage to the inside of your arteries by, say, high fat diet, high sugar diet can start to cause abrasion to the inside of those vessels and can lead to clotting. Or just having some abnormally slow blood flow. If the blood is not flowing quickly enough, then it can get stopped up and we can start to form clots that way. All right, so once we formed a clot, and this is totally normal process to do if there's been damage, eventually as we heal that damage, we want to get rid of the clot, so we need to dissolve it. And that process of clot dissolution is called fibrinolysis. So basically we take all that fibrin and we have to get rid of it. So another protein is involved here, a protein called plasminogen. That's going to help hydrolyze or dissolve that fibrin, which will eventually dissolve the clot. You also have a chemical called streptokinase, or SK. So this is a factor that's going to activate the plasminogen and help dissolve clots. So we use this to help dissolve clots in the large arteries around the heart. All right, just a few anticoagulants, which you're probably familiar with. Aspirin is considered an anticoagulant. because it inhibits vasoconstriction. So vasoconstriction is the narrowing of the blood vessel. So this prevents the narrowing of the blood vessel. It also prevents platelets from aggregating. So it's stopping those platelets from sticking to one another, which is going to reduce the chances that a clot will form. So aspirin is part of the healthy regimen to avoid getting heart attacks or strokes in people that have a tendency toward thrombus formation. Um, Heparin is another anticoagulant. So this one, heparin, we produce by your mast cells or basophils. So this is a normal chemical that's produced inside your body, but it can also be given by injection. So it basically combines with the antithrombin in blood and increases its effectiveness in blocking thrombin. So it's going to prevent the thrombin from converting fibrin to fibrin. And the last one here, Coumadin or warfarin. So this one is an antagonist to vitamin K. We haven't mentioned vitamin K yet, but it's going to be an important vitamin in your clotting process. And if we can antagonize that vitamin K from creating prothrombin, then we can stop the formation of clots. This one is a little bit slower working because it works a little bit farther up of the chain of action here. So it's happening a little bit earlier in the sequence, so that we're not even creating as much prothrombin in the first place. Okay. So a little bit of terminology here. So I've been using the word thrombus, and that's just another word for clot. So a clot is a thrombus. If so clotting that happens in any kind of unbroken blood vessel is called thrombosis. And if the clot itself stays put there, it's called a thrombus if it's not moving. And again, this can happen anytime you roughen up the inside of a blood vessel, and that can be due to bad diet, trauma, infection, things like that, can create rough spots on the inside of a vessel. And those rough spots are going to then begin to collect platelets. They can begin to form clots. Now, if that thrombus becomes loose, if it gets dislodged and it starts to move, then we call it an embolus. So the thrombus, when it's stationary, it's an embolus once it starts on the move. And emboli are very, very dangerous, right? So an embolus, once it's on the move, can get stuck in places where you don't want it to get stuck. So the worst cases like pulmonary embolism or stroke means that that embolus gets stuck and it doesn't allow any blood to pass beyond it to any of the tissues further down the line. So if it's blocking off blood supply to the brain, then that's an embolic stroke. It means that anywhere past where that clot is not getting access to oxygen anymore, and that tissue can start to die. So that lack of oxygen to tissue is called ischemia. So moving on to different blood types here. So you're probably familiar with the fact that humans have various different blood types that are programmed genetically. The most common blood groupings that we're kind of familiar with are the ABO blood group and then the RH blood group. And all these blood groups are really referring to is the presence of antigens on the outside of your red blood cells. So again, antigens are proteins. They're just proteins that the body can recognize, that the immune system can recognize as possibly being foreign. And red blood cells have these proteins on the outside of them, and that's how your body knows your own blood from somebody else's blood, right? So we're going to look at how we get these different types of blood groups and what they actually look like. All right. So again, the ABO system. This is probably the one that most people are familiar with, where you're either type A blood, type B, type AB, or type O. And that just has to do with, again, which antigens you have on the outside of your blood cells. So if you're type A, that means you have only A antigens on the outside of your cells. If you're type B, then you have only B antigens. And if you're type AB, that means you have both A and B antigens on the outside of your blood cells. If you're type O, that's just a lack of antigens. So there's no O antigen. It just means you have neither A nor B on the outside of your blood cells, right? So, and which antigens you have are going to determine who you can donate blood to and who you can receive blood from, right? So if I have type O blood, for example, that means that my blood doesn't have any A or B antigens. And that means that if somebody tries to give me blood that has A or B antigens, my body will recognize that and fight it, right? It will attack those blood cells as being foreign because it notices those antigens on the outside of the cell. So type O blood can donate to anybody, right? Because there's no antigens, it's not going to anger up anybody else's blood. Type AB can receive from anybody because he's got both antigens already, so the body's used to seeing those two antigens on the outside of blood cells. So type AB a person can receive from anybody, type O person can donate to anybody. Right. In addition to that ABO, one of the other important blood groups is the RH group. So this is the rhesus antigen that we find on the outside of some blood cells. And for this one, you either have that rhesus antigen or you don't. So you're either considered Rh positive or Rh negative. And Rh positive is more common. So this is if you're A positive, for example, that means you have type A antigens and Rh antigens. If you're A negative, you have type A antigens, but no Rh antigens on the outside of your blood cells. And this works in a similar fashion. If you're Rh positive, you can receive both positive and negative blood. But if you're Rh negative, you can only receive Rh negative blood. All right, so here's a visualization. Hopefully this will help to kind of understand what this looks like. So up here we've got type A blood, and we can see what their red blood cells look like, right? So a type A person has these type A antigens all the way around the surface of their red blood cell. In addition to having those antigens on the red blood cells itself, they also come with some antibodies in their plasma. So these antibodies are always going to be against the type of antigen that the person does not have. So in this case, a type A person is going to have anti-B antigens. And that means that if any B antigens appear in this person's bloodstream, those antibodies are going to be able to attack those B antigens because we've got these anti-B antibodies. All right, so here's what our antigens look like. Here's what our antibodies look like. A type B person over here, they're going to have these B type antigens on the outside of their cell, and they're going to come with anti-A antibodies, right? So they're ready and willing to attack any A antigens that appear in the bloodstream. A person who is type AB has both types of antigens, so they've got both A and B antigens and no antibodies, right? So there's no antibodies for either one of those because they wouldn't want to be attacking their own blood cells, and they have both antigens. Right? So no antibodies because they have both antigens. And then a type O person here, we can see there are no antigens on the outside of that, which means they have both anti-A and anti-B antibodies, right? So you can see how that makes them difficult to donate blood to, because if anyone is coming along trying to bring any of these other antigen types into this person's bloodstream, into a type O person's bloodstream, it's not going to work out well because they've got antibodies that are ready to attack any of those foreign antigens. All right, and then as far as the Rh typing goes, so the rhesus factor, like I said, you're either rhesus positive or rhesus negative. Most people, and I think in America, the stat is about 85% of people are rhesus positive and only about 15% are rhesus negative. The main problem that we find with the rhesus factor has to do with pregnancy. So if a rhesus negative mom is pregnant with a rhesus positive fetus, there's a chance that some point during that pregnancy or during childbirth, she gets exposed to rhesus positive antigens. which you wouldn't think is that much of a problem, except that Rh negative people do not automatically make anti-Rh antibodies the way that, say, a type A person automatically makes anti-B antibodies. So Rh negative people don't naturally make those antibodies, but they will do so if they are ever exposed to an Rh positive protein. So if Rh negative mom has an Rh positive baby and she does get exposed to that Rh positive antigen, then she could start producing anti-Rh antibodies, which again, usually not a problem for the first pregnancy because by the time she's exposed to that, usually she's towards the end of the pregnancy or in the childbirth stage. And so it doesn't really affect that baby. But if that particular mother then gets pregnant with a second Rh positive fetus, now she's got antibodies in her bloodstream that are ready to attack that baby's blood, right? So now there's a chance that mom attacks the new baby, or that mom's immune system attacks the new baby because of the antigens on the outside of the baby's blood cells. This can be prevented, right? So we have mechanisms now that prevent mom from forming those antibodies, but you do have to undergo treatment to prevent the formation of antibodies so that you prevent the loss of the fetus. So this could result in the loss of the pregnancy or some complications if the pregnancy is brought to term. Okay, lots of questions here. So think about these questions. What's the liquid part of your bloodstream? What are the major types of proteins that we find in the blood? What are the 3 formed elements of blood? What hormone stimulates the production of red blood cells? What is phagocytosis? What is hemostasis? What's an antigen and an antibody? And then what is hemolysis? Right, so here's your answer. The liquid part of blood is called plasma. The major types of proteins found in the blood are albumins, globulins, and thrombins. So that albumin is the albumin is good for transport, the globulin good for transport and immune function, and then the thrombin for clotting. What are the 3 formed elements? Those are erythrocytes, leukocytes, and thrombocytes. The hormone that stimulates the production of red blood cells is erythropoietin, and again, that's made by the kidneys in response to low oxygen content. Phagocytosis is ingestion of large particles through vesicle formation or cell eating. This is a thing that your white blood cells do to invading bacteria or viruses. Hemostasis is the stopping of blood flow. An antigen is a molecule, usually a protein, recognized by your immune system as being foreign. And then an antibody is your immune protein for protection against those antigens. And then hemolysis just means destruction of red blood cells. Right, so hopefully that was helpful in your understanding of blood. Let me know if you have any questions.